Global Policy Meets Interoperability, and I believe Daniele is going to be starting off with that. And then after that, we can come back together and have a bit of a more general discussion. There's quite a lot going on in the chat already, and we can pick some of those points up. So Daniele, if you'd like to take it away.
Thank you, and good afternoon, colleagues. I hope you can hear me fine. Well, we developed this paper through a very interdisciplinary team. And in fact, we wanted to reflect this in our brief presentation today. So I will start presenting and other authors will follow. I am Daniele Manzella, I'm a lawyer and I work for FAO. So we begin in this paper by observing that higher value of experimental data and genomics data in particular is achieved when they are associated to the source genetic material. And generating experimental data often implies utilization of the source genetic material. And such utilization is in fact governed by international legal agreements on genetic resources. These agreements pursue equity and fairness in the global exchange and use of such resources. Now, in a nutshell, access and benefit sharing are the two regulatory parameters of these various agreements. Source genetic material and users are identified. Access to the source genetic material for utilization is regulated through contracts, MPAs, and the benefits of utilization are also identified for equitable sharing. Now, in this policy landscape, the so-called dematerialization of source genetic material is a growing concern. Based on the observation that innovation increasingly relies on intangible components of genetic resources, ultimately data, and that such components escape the current regulations. And some policymakers think that in order to preserve equity and fairness, the scope of access and benefit sharing should be extended to data derived from source genetic material. And genomics data, provisionally defined as digital sequence information, a term that data scientists continue finding quite horrible, but it's a provisional term and it's actually the target of this initiative. Now, in praising the pros and cons of regulating genomics data, the interaction between policymakers and the genomic science community is very problematic. Not only because of radically different value propositions, but also at the nomenclature level. And in view of possible regulation, very heterogeneous data categories are subsumed under the general scope of DSI or digital sequence information. Now, in this very contrasting landscape, policy and legal solutions are slowly beginning to emerge. And given the regime complex of access and benefit sharing, meaning different international agreements, different institutions, different stakeholder communities, the current DSI debate in genetic resources global fora will likely lead over time to diversified coexisting policy and legal solutions. Now, the need to identify certain data sets as DSI or any other more accurate term that will be agreed upon in the future, as well the need to attribute data generation and use to one or another regulatory scheme, will likely call for data aggregation and interoperability among information systems. So, as authors aware of the dual and technical and social dimensions of interoperability, in this paper, which, as you will see in the next 10 minutes, is very much work in progress, we essentially propose an initial practical implementation of interoperability through the connection between genomics data and the source genetic material, based on existing information system and existing genetic resources information system. I'll stop here and Marco, over to you.
Thank you, Daniele. Okay, our solution is based on DOIs, so we have a very short introduction to DOIs for those who are not familiar with them. So digital object identifiers, DOIs, are a kind of permanent unique identifier, also called PIDs. The DOI structure is like that. There is a prefix that is the 10 dot is the prefix of the DOI inside the handle system, which is an even larger system of identifiers. Then it is a prefix assigned by the registration agency to the specific registrant authority. After the slash, there is a suffix that the registrant authority assigns and guarantees to be unique. On the right, you see example of DOIs. So the International DOI Foundation doesn't assign DOIs by itself, but relies on the work of registration agencies. And Crossref and DataCite are the two largest ones, Crossref focusing on publications and DataCite focusing on datasets. According to what our idea is about, the DOI system already offers quite a range of concepts and tools that are suitable to us. So for instance, it has a flexible metadata structure that will allow us to describe the various components of the DSI, regardless of the eventually agreed upon definition. There will be different components. There are provisions to establish relationships among DOIs and other kind of permanent identifiers. There is the PID graph, which is a initiative funded by the European Union project Freya, which basically builds a graph of relationships among DOIs and other permanent identifiers. There is an initiative called the Make a Data Count that aims at recording the number of times a dataset is downloaded or referenced. So it's equivalent to what the indexing of citations is done for papers. And in our specific context of plant data, DOIs are already being assigned to plant genetic resources, papers and publications, and to datasets. So some of the work has already been done. We would like to involve in our initiative the International Nucleotide Sequence Database Collaboration, INSDC, which is a very popular repository of DSI information. And as we can see in the screen, in the red rectangle, the source material is referenced in a sort of not fully precise way, because it's referenced as the cultivar name, IR64, which is a very popular rice succession from IRRI, and also this happens in a field that is not mandatory, so the reference to the source material may be missing altogether. Sorry, I skipped this one. This one is a screenshot of the global information system of the treaty, whereby we assign DOIs to plant genetic resources for food and agriculture. So as you can see on the screen, this is an IRRI accession, rice accession from IRRI. The DOI is the one written in green at the top. The initiative launched in 2017, and we have already signed over 1 million DOIs. We have already registered all of the CGIAR gene bank collections, but also research center, universities, national collections, and private companies. We started with ex situ and inbreeding conservation and development, but we are also going to very quickly expand to in situ and on farm. We are finalizing the guidelines for that. What I would like to draw your attention to is basically two areas of the screen. The blue screen, the blue rectangle, are links to associated information that the registrant adds to the submission to the GLIS. Instead, in the red rectangle, you see references to publication and datasets that are automatically collected by the system, taking advantage of the properties of the DOI system, because when a paper that is published references one of our DOIs, through the DOI system, we can automatically add the corresponding link to the page. And this is one of the features that we would like to use in our solution. So I will now give the floor to Pankaj.
Hi, as we were discussing a lot about the data, especially into the genomics community and the genetics community that is handling a lot of germplasm data for plants, for example, or in situ data sets for considering from across the kingdoms on developing newer varieties, improving the trait and yield and adaptation and things like that. So the data that's coming out in terms of either genes, pathways, or genetic loci or genetic markers or quantitative trait loci, peptides, sequences, and mRNA and RNA sequences and their alternative forms and things like that. So you can imagine that there is a long legacy of creating these data sets for the last 40 years or so. And the system has matured quite a lot. And so, and there are systems have been put in place. There are international archives that sync every night. And every researcher, when they publish the data on these things, objects, they have to disclose and open up their data and make their data available either through the international archives or some other things. So what they have is a constant unique accession assigned to these data sets. But when these things are published, obviously the standards keep on evolving on this format and the system, and as we generate new information. But the whole practice has always been open source and open access for these data sets. But these are also going through the versions of the data changes that are happening as the knowledge is evolving. So there is data archives, and then you have additional places where these individual information, pieces of these information is stored in dedicated resources and databases. And what we have is a distributed system. So far, everything relies on the accessions and how they are considered. So I understand that DSI was assigned to a sequence entity, but understanding that all of these entities, for example, that I'm showing on the screen, they are a small component. Not everything is having a sequenced entity in this aspect. Right, QTLs, markers, locus, pathway, gene, they could be genes that are not does not have association to a sequence. So we came up with an idea about DGO, or digital genomic object, or digital genetic object, whichever you call it. And DGO is a much broader and a bigger concept, where DSI is a component, or hardly 4 to 5, 10% of that data from the DGO would actually have kind of a sequence entity associated with it, which will go into the international archives and submitted on that side, right? So we need some mechanism how to correlate, interoperate, and build a knowledge graph that can be used by users that Nalini was talking about, but also the users that are breeding across the states, across the countries, and across the systems for benefiting on that side. So next slide, please. So what we have here is basically a knowledge graph at the back. Just ignore the round glyphs over here. So you generally have a taxon, and there have been a wider discussion about assigning DOIs to the germplasm and stock accessions. So far, on the legacy side, all these components that you're looking at, from genes, pathways, genetic maps, genomes, quantitative trait loci, or polymorphism that is detected in a genetic diversity panel, or wild accessions of the crops, they're all reported in a literature and that carries a unique DOI system. But there is only some mechanism has been put in place to identify each and every part of each and every type of these objects that you are showing in this knowledge graph, right? So what we are envisioning is that DOIs, for example, has been associated to the germplasm, but how do we connect the material that is extracted or sourced from these original germplasms and stock accessions that is represented in this thing? Plus, the second level is, if somebody else uses the same marker for his own genetic population or any other experiment that they are looking at, we should be able to track how they are being used. So one mechanism that came out was blockchain, but what we're saying is that system is already in place in many places, and it's so hard to change what is in the legacy archives that is available, right? It needs a lot of engineering and a lot of effort and mind-boggling discussion to come up to a consensus building. So what we are trying here is that why can't we use some kind of a DOI system, having each DSI or DGO assigned a kind of a DOI, where we should be able to use the citation indexing mechanisms to cover and build a knowledge graph to address some of these questions that we are talking about over here. And you can imagine some of the questions people might be asking: show me all the germplasms that have a marker that has been assigned to a drought tolerance. So this is a typical database query in a one given resource, but using the ontology framework and the citation indexes, or citing each of these object types in any number of resources, then it avoids the inconsistency of nomenclature. It avoids the inconsistency of how you store the data, but each of these objects are being cited. Although this is a different new paradigm on citation, so it will increase the number of citations in a published report, but it will be one way of looking at and extracting all the information and collecting sourcing, because we've tried various methods, including natural language processing, and it's so hard to manage all that information. So this is where I think I will end and let Marco take over.
Thank you, Pankaj. So basically, this is what we propose in the case of INSDC. The record in what the researcher will do is assign a DOI to the source material if it doesn't have already one, through a DOI registration request from GLIS, the Global Information System of the treaty, and possibly injecting ontology terms at this stage. Then this DOI, together with the actual submission of the dataset, is submitted to INSDC, as we will see later on. And then both INSDC and GLIS will be able to query the DOI system to discover and harvest information from the other components of the DSI. In the case of the INSDC, we can use an indexing system to discover the INSDC records that cite our DOIs. So basically the idea is to
jump in and say, if you could bring it towards the end quite soon.
Yeah, there is one extra slide after this. So this is what the INSDC record would look like, and this is the link to the DOI page in GLIS. Now I leave this one to Elizabeth and Brian to wrap up. Thank you.
Thank you, Marco. So what we have been discussing here is the vision of developing infrastructure and capacities to combine all information about this digital genomic object that Pankaj mentioned, and bring that back to the original source material, which in the case of the treaty would be the germplasm conserved in the gene banks or in situ on farm. So through this proposal, we would try to accommodate the definition of what a DSI is, which can be very large or very narrow. There's no common language at the moment, really agreed. So one of the objectives would be to help clarifying the language around the DSI, and this is also why we are proposing the concept of digital genomic object to make it larger. We want also, through this proposal, to develop the knowledge graph that will help researchers and breeders to extract proper information through all the linkages that we build through DOIs, and also by describing the elements with proper ontologies that already exist, like the sequence ontology and short metadata schemas. So the practical steps we are proposing is to create a specific task group that would be, of course, bringing several experts who are already engaged in existing standards and discuss the elements for a proper governance model. What would be the tools that exist or are needed for annotation of the data and the DOI allocation. And also, we would like to bring that into a concrete pilot project that we already discussed with CIAT and could be using the common beans data and the GenBank information combined with certainly also rice data. So we would have two pilots, in fact, if doable. And this task group could develop a proposal for raising funds. So our last message is we look for your feedback, of course, and expression of interest for collaborations. Thank you.
Fantastic. Thank you to Daniele and Marco, Elizabeth and Pankaj for that, which was a very, very interesting talk on a topic that I think has been quite a big, well, the topic of DSI has been a big stumbling block for quite a while in terms of governance of plant data linkage, but this new approach, I mean, the idea of digital genomic objects, I think is very interesting and something that I'm sure people will have a lot to say about. So I think what we'll do is we'll take a few questions on this specifically, but also I'd like to invite everyone to turn your cameras back on and we can transition straight into a more general discussion as we go.